welcome to week 3. This is Lobes of the Brain and Cerebellum, our second video. Thanks so much for coming back to join us this week. I hope you guys enjoyed our video last week. Let us know what you guys think about these videos. We hope you're enjoying them. We've been having fun making them for you guys. But we're going to continue to build on the foundations that we taught you guys last week with cerebellum and lobes this week. So let's get started. Some quick announcements. If you're not already signed up on our Facebook and Engage page, be sure that you are, as well as following our Instagram, just so you can be keeping track of any announcements that we're posting. A lot of you guys signed up for our email list, so we have you guys on there now, so hopefully that's how you got the link to this video or from our Instagram. So thank you for reaching out if you wanted to be on it. As well as, since you're already on YouTube right now, just click below and go ahead and subscribe to our channel so that if you want to find any of our videos, if you have to miss a week or you want to go back to the week before and refresh yourself, you're going to have easy access to all of our videos in one place if you just subscribe. So just to get started, I want to do just a quick vocab run to make sure that if we say anything that you guys aren't familiar with, that you know what we're talking about, as well as just refreshing you with some terms we went over last week. So brain is cortex. It's this big outer part that we think about when we picture a brain in our head probably, as well as so then the brainstem is going to be this thinner, more midline structure. We have our cerebellum, which is our mini brain. It's over here, and Rope is going to be doing a great job talking a lot more about it later in this video, so be sure to stick around for that. It's really important for us as chiropractors to understand it. So lesion. We say lesion a lot in functional neurology, and lesions can be pathological lesions or something that you might see on an MRI where it's something very serious, but most of the time when we say lesion in the context of these videos, we are talking about just an area of dysfunction or malplasticity, just so you guys understand what we're talking about. An ablative lesion is something a lot more on a more pathological scale than what we're talking about are more functional lesions. So VOR, vestibulo-ocular reflex, as well as VOG, so video oculography, or we call it VNG. Those are, so VOR is a reflex that we have that we're going to have 2 whole weeks on in club because it's so important for us to understand our eye movements and the power of VOR as functional neurologists. And we're going to hopefully have our VNG set up so that we can look at our eye movements a little bit more thoroughly next week. So keep your eye out for those videos in the upcoming weeks. We're going to be using those vocabulary words a lot more. Ipsilateral and contralateral. We talked about those already, but just to refresh, ipsilateral, same side. So our same side body goes to our same side ipsilateral cortex, and then contralateral ipsilateral cerebellum and contralateral cortex. So that's that pathway that we've been trying to drill home for you guys. And then pursuit is going to be an eye movement that, again, we're going to talk about next week, but just for context, it's that slow, controlled eye movement where you're just tracking a target with your eyes. And then central integrative state is abbreviated as CIS if you see it anywhere. Plasticity, we talked about this last week, but it's really just that our ability to reactivate neural networks and regrow and regenerate an area of the brain that was previously maybe dysfunctional or was just not working as well, not communicating as well. By activating those specific areas of the brain, we're able to make changes through neuroplasticity to get the results that we want with our patients. Saccades, so the pursuit was that slow eye movement. The saccade is that quick snap back. It's another reflexive eye movement that we have. And paresis is just a partial paralysis, so what it sounds like. So let's go over these big overlying rules of the brain. If you guys can understand these concepts, it'll really help you when we're going into the different lobes or the different areas of the brain to have an idea of their overall function and why they're, so that if you don't remember the exact thing that they do, you can remember their overall goal and importance in the body. So the lower and more midline a structure is, such as our brainstem, the more evolutionarily old it is. So these areas you can think of are responsible for a lot of our vital functions. The more higher and lateral areas of the brain are evolutionarily new. So like our cortex, you can think of these as areas that sometimes inhibit those more primitive aspects that in the newer areas are what make us humanistic. So why does this matter? So new areas, like I was just saying with that inhibition, new areas have the ability to inhibit older areas of the brain, as well as higher areas of the brain have the ability to inhibit lower areas of the brain. So our cortex can inhibit our spinal cord. An example of this that you guys should be familiar with is going to be our primitive reflexes that we are born with as infants. As we grow and our brain develops and matures, our newer parts of our brain that are maturing are able to inhibit our primitive reflexes, which are part of our older brain that were created for our survival is the reason we have those reflexes. So as our brain develops, we're able to inhibit those that lower function. So these are just the lobes of our brain that we're going to talk about today: frontal, parietal, occipital, and temporal. And then we're going to talk about brainstem a lot next week with eye movement, and Rope is going to hit cerebellum a little bit later. So this is just a video, and it's showing all of the different cortical pathways in our brain lit up in action together, so you can see what our brain might look like when we're actually having a thought. When we touch our arm and that proprioception goes into our brain, what that stimulus kind of looks like. So a lot of times in club, we're going to be breaking things down into these pathways and functional pathways so that it's easier to clinically use them for our patients to get them well and use these pathways that are really strongly connected in the brain. But I want you to keep in mind as we're going through it and simplifying some things in these pathways to be able to use them clinically, that our brain is so complex, and it's a really beautiful thing. So know that yes, we are going to be, when we're doing a complex movement on the right, we're activating the right cerebellum and the left brain, but it doesn't mean that our right brain isn't doing anything. So really just keep that in mind. I don't want you guys to get too simplistic when we're thinking about our brain. So as you can see, it's communicating just from all pathways in between. It's going back, information is going back and forth, up and down through all areas of our brain. So keep this picture in your mind. It's a really pretty video. If you appreciate neurology or anatomy or anything, there's some beautiful artwork showing these. Alrighty. So keep that in mind. Our brain is very complex, and appreciate that, because that is what allows us to be able to make these changes and what really makes us human and who we are. Okay, hemisphericity. We know that our brain has a left and a right hemisphere. And so when we talk about that contralateral cortex, we're talking about one hemisphere usually when we're saying contralateral. So it's crossing the hemispheres. So our hemispheres, this is kind of just a visual representation of our right versus our left. As you can tell, they're super specialized and different. Something that's pretty cool, or just a fun fact, is sharks actually have two hemispheres as well, but their hemispheres are exact copies of one another, so that while they're sleeping, they can, or just while they're swimming around, they can put one brain to rest and have the other one activated so that they can rest their brains at different times. But because our hemispheres are so different, it allows us to have this incredible capacity for complexity and specialization as a human being. It's really what makes us, that development and that differentiation is what makes us more human. So the way that these two hemispheres connect with one another and they communicate is through our corpus callosum. So our corpus callosum is a midline structure that allows our two hemispheres to stay in sync. And ideally, we want our hemispheres to be in balance and be functioning at the same level and working well together. it's natural to have a little bit of differences between our two hemispheres, such as having more of a draw to art or math or things like that. We're all going to have our strengths. But functionally, we want to make sure that our brain is working and has full capacity on both sides. We're not just leaning on one side because we can't use the other side, if that makes sense. If you guys want some more information on what some dysfunction could look like, some disorders as well as therapies, and really a lot more science behind all of this, the book Disconnected Kids by Dr. Malalo, as well as looking up any of his research. He does a lot of with neurodevelopmental disorders in children, and his work is really great. So if you want more information on this, especially if that's an interest for you, I highly recommend looking up some of his work. And so childhood milestones, something that's kind of interesting to know about the way that our brain develops is, so in the first 2 years of life, our right brain is more strongly developing. So we're looking at the big picture of the world. We're working on big muscle functions. And children, as we know, respond very emotionally to a lot of things when they're such a small infant. And then from 2 to 4, our left hemisphere starts to grow a lot more strongly. So we start to develop that more fine motor muscle control. We start to develop more of our analytical thoughts. That's when they're in their troubled toddler ages, and they're really trying to test the boundaries. So it's important that we're checking these childhood milestones, and we always hear that, but I'm going to explain why it's important. So because of the way that our brain grows unevenly, as well as just the amount that our brain is growing and developing as children, it is so crucial that we make sure that children are reaching each of their milestones and not skipping any, because if they're delayed in getting to a milestone, or if they skip a milestone, the area of the brain that is responsible for that activity could not be functioning as well, which can not be good for them as a child, but then further on in life it can cause other problems as well. There's been a lot of research showing that children with ADHD that eventually then grow out of it struggle a lot with anxiety issues later on in life, and it's hypothesized that it can be due to this imbalance. So make sure that if you are going to work with pediatric patients, they are reaching their milestones, and that's something you're having a conversation with their parents about. So how do we measure differences in hemisphericity? You can do a whole neurological exam, which is recommended. Do a thorough one, and then see if you happen to notice more cortical deficiencies on one side of the brain or the other, to give you an idea overall if there is a hemispheristic difference. Or a simple way that it's not going to be the most thorough, but just a way that you can just quickly look, would be a blind spot mapping test. So with blind spot mapping, you're going to be putting 2 dots and then having a patient cover their eye, and then around 1 dot, the doctor is going to be marking every time where the patient has whatever the object is disappear from their vision, to map out the area of that optic disc where it's our natural blind spot. And so, as we know though, when we're looking just straight ahead or at all, we don't have holes in our vision. So the way that that works, the way that our blind spot doesn't exist just in our everyday life, is through visual perception and processing. The opposite side of our brain is able to fill in for the opposite side blind spot. So if my right blind spot is bigger, we're going to say, or just think, that the left side of my brain isn't doing as good of a job in filling in that picture of my world for me, and processing to tell me what's there. Or vice versa, if it was the left blind spot, that when you mapped it, it was larger, you can think that the right hemisphere wasn't doing as good of a job at filling in that blind spot. So also, just an important note for you guys to understand when we're talking about the word hemisphericity, it's the condition to mean that there's a dysfunction or imbalance between the two hemispheres. And the way that we name and talk about hemisphericity is going to be talking about the area of decreased function. So if I say left hemisphericity, it means that there is decreased function on the left hemisphere, so you might clinically see more signs associated with right hemisphere more strong for that patient and vice versa. So how do we help? How do we affect this? So a really cool study that Dr. Carrick did in the 90s was using blind spot mapping and adjusting. So let's bring it together for our chiropractic students. So what he did is he mapped his patient's blind spots, and then he adjusted specifically on one side of the body or the other. And depending on the side of the body that he adjusted, the blind spot, when they did a post check of the blind spot mapping, the blind spot either got larger or smaller depending on the side that he adjusted. So that's really exciting, because what does that mean for us as clinicians? That is telling us that the chiropractic adjustment, depending on which side you're adjusting, can specifically target one hemisphere more than the other. So we can have extra specificity with our treatment. So that is very exciting, because that is something that no medications can do. That's one of the problems that they have in the development of medications for things like Alzheimer's or other brain conditions, because there's not a specificity. When you take a medication and it goes into your bloodstream, it's going to go all over, and that's a lot of times what creates these side effects, these unwanted things. So the fact that the chiropractic adjustment can be specific and really target a specific area of the brain if we use it appropriately is absolutely incredible. So I really want to encourage you guys to learn more about that and to learn how to use the specificity that you can have in your adjustments to use that for your patients' results to really help them and get them so much better. Okay, so the frontal lobe. So the frontal lobe is our first lobe. It's the executive. I want you guys to think about this lobe as the lobe that sets us apart and really makes us human. It's what in our brain differentiates us from all other animals, because it's that higher thinking power, that cognitive ability. So it allows us to goal set, motivation, reward, attention, to make judgment calls. It's really important in our thinking, and it's one of the last things to develop. But it's also extremely important in movement. So anytime we want to make a voluntary movement, I want to move my arm, information has to go from my corticospinal tract. So from my prefrontal cortex, which is my motor map that lives in the frontal lobe, it has to travel down into my spine to my muscle to activate that movement. So the frontal lobe is very diverse, like I was saying, with all those functions that it has control over. So we're going to talk about different pieces of it that have more specific control to give you guys a better idea of the different areas and different things that it does. So the dorsolateral prefrontal cortex is really highly involved in executive function planning. So it's going to help you decide what to do, if you should do something or not, and if so, when to do it. So that's this area right here. And then we have the orbitofrontal cortex, which is on this bottom portion right here. And so this is what allows us to live in a society and have empathy for one another as humans and really connect and act appropriately in social settings. So it's really, this area of the frontal lobe is really connected to the limbic system and the amygdala, which is highly associated with anger. And so a cool case study actually with this particular lobe is Phineas Gage. So if you haven't heard about him before, he had an iron rod that went through his skull and he survived to tell the tale. But the problem is, when this area of his brain was damaged, his movement and anything else wasn't affected. What really was shocking to all of those that knew him was he had a total personality switch. He became just a total complete jerk, and he didn't know how to have empathy for others, when to not say certain things. So that's just a cool example if it can help you remember the orbitofrontal cortex. So the anterior cingulate is really important for motivation and goal setting and having just wanting to do things. So when people are very depressed and they're stuck in bed and don't want to get out of bed and they just don't care about life, You can think of it as that sort of mentality. Those functions are what come from our anterior cingulate. So there's studies showing that this area of the brain is underactive in major depressive disorders as well as Parkinson's. So if this is something that interests you, any one of these lobes or any conditions associated with it, I really want to encourage you guys throughout this quarter, if we bring up something, to go do some research on it and find cool research articles on it, because there are a lot out there. So frontal lobe lesions, what are some things that can maybe give us a clue that the frontal lobe might be not functioning very well? So hypomemia, which is that masked face, we see it a lot in neurodegenerative conditions such as Parkinson's, Alzheimer's, things like that. Bradykinetic, which is just really slow movements, as well as noticeable spasticity in the extremities. That's more of an ablative lesion. We can think of that as an upper motor neuron lesion. That's spasticity, such as getting your Babinski's reflex back because you don't have that inhibition coming from the frontal lobe to inhibit that primitive reflex. So those are more of the motor responses that you might see, and then emotionally with the frontal lobe, you might see unacceptable social responses. Someone might be seeking immediate gratification. The frontal lobe is really important in depression versus mania, like I was just talking about with depression and the anterior cingulate, as well as mania symptoms, having that control and knowing when to do something or when not to do something, as well as expressive aphasia. So our Broca's area lies in the frontal lobe, and so expressive aphasia is when the patient can understand things, but they don't have fluency in their words. They aren't able to speak really well. So that's one condition that you might see, as well as blepharospasm. This is something definitely to look for in your concussion patients or anyone who had a TBI, because this is just going to be an involuntary blinking or eye spasm, and it's something that clinically we associate a lot with the frontal lobe. So how are we going to test this? There are some simple tests, such as the pincer finger tap test, which is when you take your index finger and your thumb with your hands spread apart and keep them at golf pulse 90-90, and you try to open and close as quick as possible, making sure that you're opening all the way up and going all the way down. And it seems like a simple test, but really no, it is important in frontal lobe function. And then gait and dual tasking. So gait itself, its movement, it's a whole brain function. So don't think that it's just a frontal lobe test, but the way that gait really can become more indicative of telling us the function of the frontal lobe is when we add dual tasking on top of it. So we're adding that extra cortical activity through asking them maybe to say the months of the year backwards, say the alphabet every other letter, something that's going to use their brain and make them think. Then we're going to activate the frontal lobe and see what that does if it alters their gait. And you can add dual tasking and adding that complexity and tasking the brain to any test we do, but know that we use it a lot with gait. It can tell you a lot. Saccades, so we're going to talk a lot about these next week, but that quick snapping eye movement, we like to say that saccades, so if I snap my eyes to the right, it's associated with the contralateral frontal lobe, so it would be my left frontal lobe. Okay, so the parietal lobe. This is our somatosensory map. It is what allows us to know where our body is in space, know where each of our body parts are, and having just awareness of the environments around us. So we can know that there's a wall there, or hey, I'm gonna trip over this step or something like that, just having awareness. And the way that this lobe gains information is through afferentation. So the brain is stimulated by afferentation, but we were talking a lot with more descending tracts in the frontal lobe with motor output. Now we're talking about sensory input to help us gain more awareness of our body and our surroundings. So this is through the dorsal columns and the spinothalamic tracts. So you can just think about how you might activate those tracts in your therapies to activate this lobe more. So this is our homunculus map of our somatosensory cortex. So this is our post-central gyrus. So right behind that motor map, we have this sensory map. And they look similar, but the proportions are a little bit different. So throughout this, you can see that different areas of the brain are mapped at different sizes, and it's based on how much awareness the brain gives that particular body part in how much cortical attention it gives it. So as you can tell, the areas that are going to be bigger, like our hands, are going to be much more sensitive to any feelings, fine touch. Our tongue is extremely sensitive, our lips. But then when you get to our neck, our trunk, our hip, look how small this is. And you would think the trunk would take up a larger portion, but it really doesn't in the brain. So keep that in mind, how sensitive an area is supposed to be versus how maybe if a person shows that they aren't insensitive to knowing where particular spots are on their trunk, it might not be as big of an issue as if they don't know where something is on their hand, because that area is supposed to be very sensitive, versus these areas you can have more leeway in the amount of air that they're allowed because the cortical map isn't as large for those areas. So what do parietal lobe lesions look like? Because it's our somatosensory cortex, if you have a lesion in one side parietal lobe, you can just have a total loss of sensation on the other side of the body. So, or more functionally, you can just have less awareness in particular areas of the body. And then you can also have hemi-neglect. So that's what this picture is showing. So the patients were asked to draw these pictures as seen on the left, which are just complete whole pictures, so a whole clock, a house, and a flower. And then their patient copy of it was just half of it. So they look pretty distorted, but to the patient, it was just a normal picture because their brain just totally ignores one half of space. They just don't know that it exists. A decrease in reflexive eye movements. I'm talking a little bit about pursuits here and VOR and OPK that those are used in, but we're going to talk a lot more about that next week. Visual agnosia is when you can see an object, but you can't recognize it. This is also going to work with the occipital lobe in this. And then we also have akinetosia, which is when you can't perceive motion. So that is an extremely important skill, and I have a cool video to show you guys of the patient perspective for what this looks like. So this is just, it's a quick 30-second video, oncoming traffic. So as you can see, normally you're just going to see the cars coming back and forth. They're smaller and they're continuously getting bigger. Versus a person with this condition is just going to see them, and then all of a sudden they're going to see the car right next to them. It's going to be far away and then right next to them. Or when they're pouring a drink, they're just going to see it at particular levels. So it just looks like the video is jumping, but that's showing just what their perspective looks like, which would make it hard for them to do simple tasks such as pouring a drink because they can't see the movement of the drink as the glass is filling up. So think about that, it's pretty cool. So how do we test the parietal lobe? We use point localization. So that is when you're going to touch an area of the body and have the patient, with their eyes closed, try to match where you touched. And so this is what I was talking about earlier with that leeway that they're allowed to have. So on the hands, if they're an inch off, that's more significant than if you're up on their shoulder if they're an inch off. So you want to think about the areas of sensitivity. You want a smaller degree of error of where they're landing, where they think you are, versus if you were touching on their stomach or something like that. They might be allowed to just have a little bit more leeway. So it's all side of the neurotip, Q-tip and finger. That's just going to be our dorsal column stimulation, spinal thalamic tract, blind spot mapping, and graphesthesia. So blind spot mapping, just so you guys know, we're not going to show you the whole thing because it does take a lot of time, but there are great videos on it if you want to look it up. But basically, the idea for you guys to understand that we want you to take away is if you did a blind spot map and you drew a line in the middle of the dot that you had them, so the blind spot map was a circle around, around the dot, then you would know that the bottom portion, so if the bottom portion of the blind spot was bigger, then you're going to think that that might be more of a parietal lobe lesion on the contralateral side. So just for your awareness, blind spot mapping does have to do with you can use it in context for parietal lobe as well. Graphesthesia is one that we classically learn in school. So it's going to be where you're just drawing a letter or a number on a patient's hand, having them try to recognize what you're writing while their eyes are closed. All right, so let's go to our first breakout. And so grab a partner if you don't already have one, and let's go practice. All right, guys, time for our first breakout. So I hope you guys grabbed your partner. Mine is Rope. Thank you very much. You could also try to use someone like Chester. I don't know how to work, but you could try. Just practice. So we're just going to start with our frontal lobe test. We're going to start with the pincer test. So take your index finger and your thumb together, keeping your hands open, opening all the way up, opening as quick as possible. And it looks pretty good. So now that we've seen that, you're going to be looking for, while they're doing this, if they have hesitations in their movement, if one side is quicker than the other, or if they're not opening all the way, they're just doing a small movement like this. You're just going to be looking for really just any asymmetries between the sides to give you some clues into their frontal lobe. And it seems like a really simple test, but I really want you guys to know that this is important clinically, and it's so easy and it doesn't take anything that why not do this on all of your patients, because it can really show you some insight. So now we're going to tax the system a little bit more with dual tasking. So we're going to add extra cognitive thinking to activate that frontal lobe and see if it changes any of his results. So I want you to do the pincer test again, and this time say the months of the year, every other, going backwards.
Going backwards.
Starting at December.
Okay. January, November, September, June, March.
I didn't even know that we came so far. Okay, well, yeah, so I think I gave really unclear instructions, but it was really hard, as you could tell. It was taxing his system, and not only because he was trying to say the months of the year in a really weird order, but especially because he's also saying them in English and not Finnish. So he did a great job. So now we're going to look at gait analysis. So as soon as your patient is walking in the door to your office, you want to be looking at their gait. Even just, it's really fun. People watch on the street and start watching people's gait to see if you notice anything. As chiropractors, it's something really important that we hone in on our observation skills to see the things that our patients might not be able to tell us. So we're just going to have you give us your best catwalk strut, just back and forth, so we can see what his normal gait looks like. So we're paying attention to arm swing, head tilt, rotation, if he's leaning to one side more or the other, heel walk, toe walk, all of it. And so really, when we're saying the frontal lobe has to do with movement, and so gait is movement, but it's really using our whole body process and our whole brain when we're just walking. Hopefully it isn't a very taxing thing and it's not using a lot of effort from our frontal lobe when we're just normally walking. So the way that we're gonna make this a little bit more frontal lobe is to add in that dual task and use more cognition and see if that changes anything in his gait. So let's have you subtract 7 from 100
All right.
while you walk.
100, 93, 86, 79, 72, 73, 72.
All right, that's it, you're good. So what you want to be looking for with this is sometimes a patient's gait will actually get better when they start doing this. So if before they were kind of just like not moving one side of their body, maybe when you add in this brain task, they start moving actively and normally on both sides of their body. So maybe if their central integrated state was low and their overall firing and neuronal networks weren't activated properly, they weren't doing very well, when you add in this extra task, it brings them up so that they can move in a normal and fluid-like motion. So, but sometimes you'll see that actually when you add in an extra task or something complicated, it will start to break down their motor function. And so this is really going to be exposing all of our subcortical areas of the brain that when you break down that frontal lobe and that inhibition, you're going to see other areas of the brain come through. So now let's go into parietal lobe. Now we're going to do graphesthesia. So just put your hands out like this, and I'm going to be drawing a letter on your hands. So just close your eyes and tell me what it is.
A, C, do it again, please. S, B, R.
Okay, good job. So one quick tip, so whether you're drawing numbers or letters, make sure when you're drawing on the patient's hand that you're drawing upside down for you so that it's the right side for the patient as if they would normally read. And then another thing, if you're choosing to do letters, I personally like to use uppercase letters because all of us learn to draw our letters a little bit differently sometimes. So maybe when you're drawing your letter one way on a patient's hand, it might not make sense to them, not because they don't understand the letter, but because they don't recognize the way you drew your hand. So that can be complicated, but just try to keep it simple. I like uppercase. So finger to nose. There's so many different versions of this test, and it can test a lot of different systems. So there's finger, nose, finger. When you're putting your finger out here and you're asking the patient to go back and forth, I can have the patient move around and follow my finger. And so these are testing all kinds of different areas of the brain, but I don't want to go too much into parietal lobe with this because a lot of those tests are actually looking more into cerebellar function. So the way that they are parietal is to know where your fingers are in space and knowing where your nose is in space. And if the patient has their eyes closed, knowing which finger the doctor might touch. So those are the parietal aspects, but I'm going to let Rope go more into that during our cerebellum breakout later. And then point localization. So just have you put your arms out and close your eyes. And I'm going to touch on your arm, and I want you to take the opposite hand and touch where I touch. Okay, so there's different levels to this test, and as you can see at the beginning, I was keeping my finger on so that he had more information. It was a lot easier to know where my finger was. He just kind of had to find it. And then at the end, I started just doing a light touch, and he had to remember where my finger was a few seconds before. So as you're using this test as a therapy, you can change the complexity of it. But when you're just looking at it to assess function, what you want to look at is, so down when you're assessing, say you touched a place in the hands or the digits, like I talked about earlier, you want to make sure that they should be more sensitive, so they should be more accurate in these lower areas. And then as I go up his arm and into his trunk or shoulder, he might not be as accurate, but it also might be because there's not as much brain mapping for these areas. So just keep that in mind. And if they're like a few centimeters off, no biggie. If they're like an inch off up here, not that big a deal. But when you start seeing consistently that they're really off in a particular area, or if they're really off down in an area that should be a very small distance to where they have that freeway to be off, then that's where you want to be more concerned or maybe do a therapy in that area. So the cool thing with a lot of our tests that I didn't mention with frontal lobe, but I want to mention, especially with parietal with this test, is the test can also double as the therapy. So the way that you can use point localization to help remap a certain area of the brain is you can have the patient, if they're missing a certain area, you can touch that area, have the patient open their eyes, and then look and see where it is and cognitively process where that area is on their body when you're putting in that afferent information touching it. And so just doing a simple exercise like that can help remap their brain. So if you have a friend who is always hurting that right ankle, check out their map of that ankle and see if maybe you can help them fix it so that they don't keep re-injuring that area, because maybe it's the mapping issue instead, or maybe it's coordination, but it could definitely just be a mapping issue for them. All right, so try out some therapies, and see if you can make any changes. All right, that was a great breakout. Thank you to those of you who participated with us. And if you just watched and laughed along with us, that's cool too. So we're gonna come back with our temporal lobe, which is our auditory and perception processing center. So auditory information. So we think about this as the main role of our temporal lobe, but I want you guys to look at this list and understand that our temporal lobe does so much more. It's really involved with our limbic system, which is responsible for a lot of our emotional drives. It's involved in the recognition of familiar faces and objects, as well as long-term memory and processing speech. We have our Wernicke's area and this area of the brain, as well as word and number recognition, as well as hallucinations are associated with this area of the brain. So look at this list and think clinically how your patients might present, what some of their complaints might be if they're experiencing difficulties in the temporal lobe. We want you guys to understand the ideas of what each of these areas of the brain do, so that when your patients come in describing certain symptoms or complaints that they're having, you can associate it with particular parts of the brain, just to keep that in mind before you do your testing, to pay attention to those areas. So if you think about it, and then once you understand the functions of these lobes, you can start layering therapies, which gets to be really exciting and cool because you can really turn anything into a therapy. If you think about just listening to music, you're going to be getting that auditory processing. Whether you're doing high or low frequency music, and the specific key that the music is in, you can be affecting one side of the hemisphere, one hemisphere more than the other. So you can be really specific in what you're trying to do. So maybe you're going to have your patient listen to music and you're going to adjust them while you are having them look at the picture of a loved one and having them recognize the face of that person, or you're having them recognize an apple, panda bear, things like that, while you're doing your therapies. And really you can start layering how you would activate particular areas of the brain to get stronger stimulation and results as the patient is able to increase the tolerance and metabolic capacity that they have. So some temporal lobe lesions that we might see is Wernicke's aphasia or receptive aphasia. So it's kind of like the opposite of Broca's. It's when they can speak and form some sentences, but they don't understand words. It's that processing. They don't understand messages. Prosopagnosia, which is when they can't recognize familiar faces. So like I was saying, if you wanted to use a patient like this, use a therapy with them to stimulate this lobe, maybe you could have them do an eye movement. And we're going to talk about how to use eye movements and how exactly that is going to be stimulating the brainstem and the brain more next week. But think about doing an eye movement and then having them do an eye movement to a picture on a phone of a loved one, or so they can go back and forth between faces. Or when you're doing something like an optokinetic strip, which is a red and white stripe kind of cloth that you can use to go back and forth to show a reflex that we have. If you can use the normal stripes, but if you wanted to add in that extra layer to make it specifically more of temporal and get more cortical activation, you can maybe have it be objects that are passing through the patient's vision and having them recognize and point to, okay, this is a panda bear, this is a butterfly, this is a koala, and having them recognize these different things can make it more temporal. And then we have memory loss because of our long-term memory. So that's a common one that you guys are going to see. So how do we do this? So again, our blind spot mapping. So like I was talking about with the parietal lobe, if you have the blind spot on the bottom part that's bigger, we're going to think more of a parietal lobe lesion. If you see a bigger half of the blind spot, so the visual perception isn't being filled in as much on top of the dot, so on the upper field of vision, then we're going to think of that more as a temporal lesion. We are also going to do our finger rub test that auditory perception, as well as sound localization. So just like we have a somatosensory map of our body, as well as that motor map in our brain, we also have an auditory map to help us know where sounds are coming from. just in our environment. And so with sound localization, we're going to teach you how to test that and then maybe how to rehab that as well using the same test. So the occipital lobe is our visual processing center. It's going to help us with color discrimination, processing vision, and it's going to give us that map of our visual field. So like I was just saying, we have an auditory map, we have a visual map. We have all of these different maps constantly in our brain to help us stay aware of our environment and know what's going on. It's part of just our survival mechanism to really be aware. So that's the functions of the occipital lobe, and then any kinds of lesions that we're going to see with our occipital lobe are going to be visual deficiencies or loss of visual function, visual agnosia, like I said earlier. Is also it's parietal as well as occipital. So how we're going to test this, there's many different ways to test the occipital lobe, as well as all of the lobes that I'm teaching you tests for. There's a lot of different ones, but we're just choosing very specific ones that we think are some of the best tests. So one of the best tests for occipital lobe is confrontation, which is when you're going to stand behind or in front of the patient. Hopefully, I like to stand behind, and so you bring an object into view, you have the patient tell you as soon as they see it, and then have them tell you if it's moving or not moving. So, and you want to go in all areas of vision, so do both sides to see if there's a difference in either side, and then tell them if it's moving, if it's not. But make sure that you're not just going like moving, not moving, moving, not moving, so you can keep the patient on their toes. This is a chart that we, with visual field defects that we see a lot in school. It's very common. I just want to draw attention to this lower half, which we see these type of deficiencies in occipital lobe lesions. But actually, this will be hard for you guys to see, so I'll go back to this slide. So just something clinically that I want to teach you guys if you want to use occipital lobe stimulation to stimulate a particular lobe of the brain more than the other. When you're using visual stimulation, you can use the where you put visual stimulation to stimulate a particular lobe more than the other. So I'll give an example because I don't think I said that well. So if you wanted to use light therapy, so you wanted to shine a light in a patient's eye to get visual stimulus to activate their brain. If you shine a visual stimulus more medially, so nasally, you're going to be activating more of the contralateral occipital lobe, or more of the contralateral cortex is what we like to say. Or if you're shining a light more temporally, so more towards the peripheral of their vision, you're going to be affecting more ipsilaterally the cortex. So if you have your patient look over to the right, and you have a little bit of light coming nasally into the left corner, as well as more peripherally to the right field of vision, then we're going to think that we are stimulating more of the right cortex. You can think of it that way, but we're going to go a lot more into eye movements next week, but this is just a quick tip for you guys to look at and see if you can make any differences with this. So more medially, contralateral, more temporally, you're going to think ipsilateral. Great. So now we're going to go to our second breakout. Okay, guys, so we're back for breakout 2. So if your buddy left, go grab him again because you're going to need him. So real quick, can I just have you sit down for our temporal lobe? So first, we're just going to do our basic finger rub test. So can I have you close your eyes? And I'm going to rub my finger in your ear and tell me when you can hear it. And tell me when it goes away.
No.
Okay, so keep a mental note of where that was, and then test the other side.
Here we go.
Tell me when it goes away.
No.
Okay, and so if your patient has amazing hearing and you find the ear all the way out here, one thing you can do if you want to make the test a little bit quicker and make sure that they're really hearing it and not just trying to impress you, so you can actually stop rubbing your fingers and make sure that they notice that the rubbing stopped. So next we're going to do sound localization. So just have you look straight ahead with your eyes closed, and I'm going to be snapping in different areas, and I want you to take your hand and point to where you think the sound is coming from, okay? So close your eyes. It's pretty good. So with sound localization, you want to see if they are pointing in the right direction, so really towards where you are actually snapping. And if you start to notice one side that they're just totally off, you may want to look at rehabilitating part of that auditory map that they have. So now into the therapy aspect, how would you help remap it? Just like similar to point localization earlier, now we're going to do sound localization, so you can snap and then have the patient open their eyes and look where you actually are, so that, and have them, even if you want, bring their finger towards it with their eyes open to help bring that cognition in to remap that, that sound map for them. And then now we're going to go into occipital lobe. So occipital lobe, vision, but one of the best ways to do this is confrontation, which combines our visual inferentation, as well as we're going to assess if they can detect movement. So what you're going to do is you can stand in front of your patient. I'm going to stand behind so you guys can see. You're going to have the patient just look straight away forward, and then you're going to bring objects into the patient's field of view. Slowly have them tell you as soon as they see the object, and have them tell you if it's moving or staying still. And you can do this one side at a time, or you can do both sides at the same time. So let's get started. Just look straight ahead and tell me what you see.
Moving on the left. Yep, moving.
Okay.
I see it, it's not moving. I see it, it's not moving. I see it, it's not moving. I see it, it's moving.
So you want to make sure that you're going in a variety of different areas. Hopefully it's nice if you go in kind of like a clock area, but you don't want to just go moving, not moving, and try to spice up the pattern so your patient doesn't know what to expect or doesn't catch on, so you can really get an accurate test. But that's it for our cortex breakout. So now I'm going to hand it off to Rope to teach you guys about the cerebellum.
All right, thank you. All right, cool. So we're going to wrap up our conversation of lobes of the brain with the cerebellum. So the cerebellum is a massively important structure for us chiropractors and as functional neurologists. So when we talked about the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe, all the functions of those lobes and whatever is happening in the cortex has to be monitored by one structure or one area of the nervous system, and that's going to be the cerebellum. So the cerebellum basically is this massive functional processing unit that takes all the afferent information from your body and all the efferent information from your brain, and it compares those two to make corrections if there's any errors in the system. That's how the cerebellum basically works. So if there's two words you have to discuss the cerebellum or describe the cerebellum, it would be error correction. And then classically, when we talk about the cerebellum, if my mouse will work, there you go. We talk about the cerebellum of the ABCs for their function is accuracy, balance, and coordination, okay, in regards to movement. So in order for me to perform the most accurate movement possible, just for example, me picking up something from the table, I have to have information from the visual system, the vestibular system, and the proprioceptive system. And what the cerebellum does, it'll take in all that afferent information from these different systems, and each of these systems are telling you a certain story. And if that story doesn't match the other system's story, then there's a mismatch. And then the cerebellum is going to be like, hey, we got to fix this. So for instance, the cerebellum might modulate the vestibular nuclei in the brainstem to say, hey, this is not what we got from the visual system. Let's make corrections into this system. That's how it basically works. And then it has a lot to do with balance. So with a lot of cerebellar dysfunction, you see balance issues because it's so related and connected to that vestibular system as well. And has a lot to do with muscle tone that we're going to be discussing in the upcoming slides. So it has a lot to do with timing and synergy of muscle groups to make fluid movements. Okay? Then also coordination and cognition. And cognition is something we don't really talk about as much. I'm going to get a little bit more into that in the upcoming slide. But in regards to movement, it also fine-tunes motor programs. So this is where practice makes perfect. So your cerebellum has these pre-programmed or these learned movements and patterns that is stored from your life, entire of your life, from development to your adulthood. And picking up a pen or picking up a bottle from this table is fairly simple, and it doesn't take a lot of cerebellar activity to perform that movement. But if you're doing something more complex, such as starting a new sport, at first you feel like, oh, this is really hard. I'm not able to do this. Why does it feel so hard? It's because you're not used to doing those movements. And this is where practice makes perfect. The cerebellum fine-tunes those motor patterns so you can produce those movements even in kind of subconsciously. Okay? So something I want you to take out of this is that there is no direct motor output from the cerebellum. It just modulates descending pathways. And that's really important. And we're going to be talking about that a little bit in the upcoming slides. So just a few fun facts. Even though the cerebellum is only about 10% of the total mass of the brain or the nervous system, it houses about 50% of the total number of neurons. Okay? And the average neuron has about 10 to 20,000 synapses, but an average cerebellar neuron has 150,000 to 250,000 synapses. And that just gives you a little bit of perspective how freaking important this system is for fine-tuning and making sure that everything is organized and coordinated in your brain, so you can have the most appropriate motor output possible that's possible at that current moment. Okay? All right. So then, this is a lot of text, but it's such an important point that I want to read it out for you. So recent evidence has shown that the cerebellum's contribution to control of all brain functions, especially cognitive and behavioral controls, may be just as great as its control over motor functions. So cerebellum may in fact be the key to normal cognitive and emotional development of the brain, and is in fact the key to learning anything, whether it is motor or cognitive. So this is something that we don't really talk about in school as much, is the cognitive aspect of the cerebellum. So we know that the cerebellum is able to modulate different functions of the cognition, such as the amount you can think at a given time, so this cognitive consistency. And we know that this from research done by Dr. Schmallman. He's the main guy. I have one paper here that I'm going to show you, but he's the #1 guy when it comes to cognition and cerebellum. And we know that the cerebellum affects the cognitive consistency, cognitive capacity, and cognitive appropriateness. So basically what it does is it's increasing, or it's making sure that the more appropriate side of your human function is working correctly. So it works really, really strongly with the frontal lobe that we talked about last week already. Okay? And now I'm going to show you just a couple research papers on why we care about the cerebellum so much, not just the motor aspect of it. So this is one paper that concludes saying that the structural and functional imaging results indicate that the cerebellar dysfunction may also play an important role in pathology of ADHD. So it has a lot to do with these neurodevelopmental disorders as well, and their etiology and their pathology. And we know that this could be in relation to the fact that the cerebellum has such strong and dominant connections to the contralateral brain, and the development of those cortical structures, which are so important for attention and cognitive abilities, executive function, and being able to read and interpret words. So we know that the cerebellum is absolutely crucial. Then we have another study saying that the cerebellum is one of the most consistent sites of abnormality in autism spectrum disorder, and cerebellar damage is associated with increased risk of autism symptoms, suggesting that cerebellar dysfunction may play a crucial role in the etiology of autism. So yet again, another neurodevelopmental disorder here, and we know that there's more and more kids getting diagnosed with autism and ADHD. So the cerebellum is something for us chiropractors to research more on, and to try to understand the system better, because we know that we can help these kids. Okay? Then this is the paper I was talking about, the Cerebellum and Cognition by Jeremy Schmallman. He's the #1 guy when it comes to cerebellum and cognition. If you're interested in this, go ahead and read this paper or any of this work. All right, so here's a picture of the cerebellum. We all know that it's in the base of the skull, and it's also called the little brain. It's such an important structure, as you already now know. And the reason for its appearance, the way it looks like, I don't know what to call it, but why it has those folia and those folds is because that gray matter on the outside of the cortex has folded together, okay? And then inside of it, you have this white matter tracts. And in the white matter, you have those deep cerebellar nuclei that we're going to be talking about. But what I want you to take right now is that the gray matter, the cortex of the cerebellum, is going to contribute to those cerebellar nuclei that are deep down in the white matter, okay? And anything that leaves the cerebellum, or any excitation that comes out of the cerebellum and modulates other parts of the nervous system, is going to come from those deep cerebellar nuclei. So that's why it's a really important concept I'm going to give you right now before I even talk about the anatomy. But so then we talk about anatomical, phylogenetic, and functional divisions of the cerebellum. The reason why I wanted to put all these in here, because there's so many similar terms when it comes to describing the cerebellar anatomy. Like when you read about it here, spinocerebellum, archicerebellum, cerebrocerebellum, neocerebellum, the flocculonodular lobe, like there's too many of these terms, and like, what the hell are you talking about? You know, we gotta be able to, you know, figure out which part of the cerebellum are we actually discussing at the moment. So when we talk about anatomically, this is the best picture for that. Anterior lobe will be the one on the yellow, then we have the posterior lobe, the green, and the flocculonodular lobe in the blue. When we talk about phylogenetically, from oldest to the newest part, the flocculonodular lobe is called archicerebellum, and the name kind of gives it out for you. And then next we have the paleocerebellum, which is usually considered to be part of the anterior lobe. And then we have the newest part of the cerebellum, which is more the lateral zones and the more posterior parts of the cerebellum, which is called the neocerebellum. So again, the name kind of gives it out for you. So those are just some different terms that you have to be able to distinguish and understand that they're just talking about the same thing, basically. Then we have the functional divisions that I'm going to be using. I'm going to be using those terms because we're a functional neurologist here, so we want to understand what these areas of the cerebellum are responsible for and how can we treat them. So first one, vestibulocerebellum. So the name already tells you, vestibulocerebellum has connection to the vestibular nuclei, and it modulates the vestibular system and then back to the cerebellum with the flocculonodular lobe. Then we have the anterior lobe, the paleocerebellum, or the spinocerebellum. So the spinocerebellum is the functional name for this, okay? Then we have the posterior lobes, which are associated more with the lateral zones, and that's called the cerebrocerebellum. Okay, so we have cerebrocerebellum, we have the spinocerebellum, and then the vestibulocerebellum. Okay, so then I'm going to break those down a little bit for you. So the vestibulocerebellum, obviously we already know it has something to do with the vestibular system. So what it does is that this flocculonodular lobe is really connected to the vestibular nuclei in the brainstem. And it receives afferents from that vestibular nuclei, and it fires back to the vestibular nuclei to modulate it, to make sure that the vestibular system, the central vestibular system, is doing its job correctly. Okay? Some dysfunctions you might see in the system is such as truncal ataxia. You could see eye movement deficits, some problems with eye movements that we're going to be talking about more in the VOR weeks, week 4 and 5. You might see nystagmus. So nystagmus is when your eyes drift away from a target and then jump back to this target. So you have a slow movement and a fast movement. Okay, and there's different kind of nystagmuses, which we're not going to get into right now. But all those have to do with the function of your vestibular system, your eye movements, and also your posture, okay, and your balance. Then we have the spinocerebellum. So the spinocerebellum is associated with the midline structures of the cerebellum. This is another great picture. And the spinocerebellum also has the vermis. And if it helps you at all, we know the vermis, it's kind of like a mnemonic, but you can think about the V as vestibular, E as an eye movement, and S as spine. So the spinocerebellum has to do with all those things as well. Okay, it makes sure that there's postural stability and that the spine is working correctly, and there's overall stability in those midline structures. Okay, and then we're going to be talking about the nuclei that go with these areas of the cortex in just a bit. Okay, hopefully I haven't lost you yet. And then we're going to talk about the cerebrocerebellum. So we have the posterior lobes, the more lateral parts of the cerebellum here. And that has to do with more of timing of movements and initiation and planning and coordinating upcoming movements that you're going to perform. And it has a lot to do with cognition as well. So these lateral zones are the ones responsible for integrating for cognition. Okay? And some of the dysfunctions you might see in this area would be dysmetry of thought. So you're not able to coordinate your thoughts accordingly. Some of the things you might see, some limb dysmetria. And then with the spinocerebellum, with dysfunctions, you might see some truncal ataxia. You can see other forms of ataxic walks where you're going to have a white-based gait because the patient has bad balance, so they don't want to fall. So they tend to walk like this, and then they have titubations like this. It's hard to demonstrate that, but if you want to see what a cerebellar ataxic gait looks like, go to YouTube and Google it. There's plenty of examples in there. But that's kind of like the divisions of the cerebellum right there. So what I have another actually slide that'll give you a little bit of like a mind map of what's going on here. So we started off with the vestibulocerebellum, cerebrocerebellum, and the spinocerebellum. Okay, those are our functional divisions. And then all these parts of the cerebellum are associated with deep cerebellar nuclei that are going to give the output of the cerebellum. Okay, so here we are. We have a cool way of remembering these. The way it goes is fat guys eat donuts. So fat as in fastigial nucleus in the midline, you can see right there. Then we have the interposed nuclei, which consists of globose and emboliform, and then we have dentate. Okay, so fat guys eat donuts. And if it helps for you to make sure we're going from medial to lateral, the fastigial, the fat, means that this guy has a big fat belly, so it's in the midline, and then you start going laterally to the interposed nuclei and then the dentate nucleus. Okay, and all of these nuclei have kind of like their specific functions or more biased functions over the other functions. Okay, so the fastigial nucleus has a lot to do with eye movement and the midline stability of your spine and your vestibular system. Specifically, the rostral fastigial nucleus has a lot to do with trying to figure out where your head is in space and in relation to gravity. Okay. And then we also know that the fastigial, because it's in the very midline, it has to do with spinal stability, like I already mentioned. And then interposed nuclei, once we start going more lateral, the globose and the emboliform, what they do is they fire really strongly into your red nucleus, into your mesencephalon, to produce these rubrospinal tracts. And this is how you get muscle tone. They also fire in the verticulospinal tracts that also produce muscle tone. And this is one of the reasons why if you have a lesion in the cerebellum, you often see these hypotonic presentations on one side of the body. And this is because these connections from the cerebellum to these different nuclei in the brainstem that are responsible for producing muscle tone on one side of the body. So the main thing you want to take out of this is that when you have a cerebellar lesion, you usually find the findings ipsilaterally to this body. Okay, and globose has more to do with the proximal stability, so it controls your shoulder joints. So rapid alternating movement doing rapid alternating movement here is different than doing it here. So if you do it here, it's more for the emboliform, and E for elbows. That's how you can remember it, and then you can test it here. Then we have donuts or dentate, and dentate D for digits. So we're getting more lateral, like this. So you can do this test. We call this the piano playing test, and you can see if there's a difference in one side or the other. And this has to do with this is the most lateral part. And the reason why we're looking at the dentate nucleus and the piano playing, the finger dexterity, is because the dentate nucleus fires strongly into the cortex, which forms the lateral corticospinal tract, which are responsible for these fine-tuned movements and these more accurate, specific distal limb movements. Okay? All right, so hopefully you got that. It's a lot of information. Now we're going to go into just a review. This is kind of putting everything together. So we talked about the cerebrocerebellum, the more lateral zones, okay, the newest part of the cerebellum, and that is associated with the dentate nucleus for premotor cortex, for motor planning, like I said earlier. And these are pathways to the upper motor neurons. So like you can see, the cerebellum, it doesn't really have direct motor output by affects the descending pathways. Okay, so here's our cerebrocerebellum associated with the dentate nucleus, and dentate nucleus coming in affecting the premotor cortex for motor planning. Then we have a spinocerebellum. So the spinocerebellum was the intermediate zone. It was the midline. It's connected or associated with the inner post and the fastigial nuclei, and those fire into the red nucleus and the cortex as well for motor execution. Okay, so this is the more direct connection of our muscle tone from one side of the body to the other. Then we have the vestibulocerebellum, which doesn't have any connections to these deep cerebral nuclei. It only connects to the vestibular nuclei, so it has a really strong connections to modulate the vestibular responses. Okay, and the vestibular nuclei then fire in the lower motor neurons in the spinal cord and in the brainstem to create these vestibulo-ocular reflexes, such as we're going to be talking about week 4 and week 5, which are absolutely important in creating spine stability. And this is something really important we have to look at when we're scanning our patient's spine. Okay. Then we're going into clinical signs. So what do you see when somebody has a cerebellar lesion? What are the most common things to see? Well, I listed a few of them. There's a couple more coming after this. So number one of the things you see is abnormal stance and gait. I kind of touched on this a little bit already. So you see this wide-based truncal tremor, stumbling gait. So you see the patient walking really wide like this. And then we know that alcohol affects the anterior lobe of the cerebellum, and this is why you often see cops doing these tests in the middle of the night saying you're not intoxicated by doing this tandem gait. So when you do the tandem gait, you're more prone to lose your balance, and we know that the anterior lobe has a lot to do with balance in the cerebellum. Okay. Then we have titubation. So that's basically when you have this wide-based gait, you have these titubations from anterior to posterior to left to right. Just because you don't have coordination, you don't have that muscle spindle feedback coming into the cerebellum and having that proper sensitivity in that system, letting your brain know what's going on. Okay, errors in movement. We have initiation, braking, direction, velocity, and muscle synergies of movements, especially seen in finger-nose testings and rapid alternating movements. Then we have dysmetria. So dysmetria, if you don't know what it means, it's just basically a lack of coordinated movement, and usually associated with undershoot and overshoot of a target. So if I'm trying to touch, let's say a doctor is giving me a finger here to touch on, and I'm supposed to do this, I might overshoot the target, I might undershoot the target, or I might be just all over the place with the tremor at the end. Okay, so dysmetria is not being able to hit the target with possibly with undershoot or overshoot and a lack of coordinated movement. Okay, but then we also can have dysmetria of thought, and this is what Dr. Jeremy Schmaumann has discovered a lot in his research. So basically what it is, it's a mismatch between reality and perceived reality, or inability to put thoughts together. So usually one good example of this could be not a lesion, but how you can see it in everyday life when you're studying for, let's say, eight hours in straight, and you start feeling like, oh, I can't take in any information, this is not making any sense, and you're gonna go for a walk outside, and then you come back and like, oh, this makes so much more sense. It's because you created movement, and cerebellum is this novelty-driven based system that it wants movement and all this afferent information coming in so that it can function. And then we know that your cognitive abilities will go up with movement. Okay. DDK, so this is the positive for rapid alternating movement, dysdiadokinesia. Then we have dysarthria, which is basically this hoarseness of your voice, abnormal tone, hypotonia, and pendular reflexes, and you'll see this ipsilateral to the side of the cerebellar lesion. Okay, so hypotonia just means that you have less muscle tone, there's less alpha and gamma motor neuron activity happening on that side of the body. And you can feel that with palpation, you can feel that with different range of motion tests. And then for the pendular reflexes, so you hit a tendon reflex or a myotactic stretch reflex, what you'll see is these pendular actor movements after you hit the reflex. So for example, if I would hit my patella here, you'd see this pendular movement after I hit the reflex. Okay, so then let's go to the next slide. Then we have abnormal rebound tests. So classically, you see videos on YouTube where they have a cerebellar lesion patient, and the doctor will hold their hand And when they release the hand will shoot over to the other side. They can't stop the movement of the hand. Now, when you do this with your patient and you think that they might have a cerebellar lesion, don't make sure that they're not hitting themselves. Okay, that's really important. But basically what it comes down to is that we have this feed-forward efferent copy and these feedback systems in the cerebellum. We have an efferent copy, meaning that we have a predicted feedback of a certain movement. If I'll go ahead and pick up something from this table, I already know what's supposed to happen. My cerebellum and my cortex are communicating together, saying that, hey, this is what I expect to happen. Then you have the actual feedback coming in. I grab this object, and if that went well and the predicted feedback matched with the actual feedback, we're happy. But with the cerebellar lesion, this predictive feedback mechanism might not be there, and we don't even know where this movement is supposed to stop. Okay? That's just one way of explaining that. Then we have abnormal ocular motor control, gaze-evoked nystagmus, skew deviation, saccadic dysmetria, and post-saccadic drift, which we're going to touch on later on in future club meetings more. All right, so let's go ahead and test some of these cerebellar findings and see what we can find. I'm going to have Rebecca come here, and we're going to do all these tests, and I hope you can do this with your friend or your partner as well and see what you can find. Cool. All right, cool. So breakout time. Now we're going to go ahead and test the cerebellum. There's a lot more tests for the cerebellum, but these are the ones that we feel like are the easiest and the most essential that you can do without any equipment. Okay? So first we're going to start it off by looking at your posture. So the classic cerebellar posture, if you have an actual cerebellar lesion, is often internal rotation of the hand and internal rotation of the leg like this. And when you look at Rebecca here, she doesn't have any of that, okay? And a good way to look at the internal rotation of the hand is looking at the knuckles. So how many knuckles do you see on this side versus on that side? So if you see more knuckles here, it's more internally rotated. So you kind of want to use these cues to help you with your observations. Okay, next we're going to do tandem gait that we already talked about. So tandem gait, what I'm going to have Rebecca do is go ahead and take a step forward like this over the other foot like that. Perfect. And you want to make sure that the patient's not falling down. So you want to be next to them and making sure that they don't fall. Because if you're expecting a cerebellar lesion, most likely they're going to have trouble doing this. And a clinical thing that you want to take out of this, if you're just looking at for functional disconnectivity in the system, often they tend to fall to the side of the cerebellar dysfunction. So Rebecca might fall to the left every time he's trying to take a step, okay? Or to the right. There you go. So next we're going to do Romberg's. So Romberg's is classically the dorsal column test, checking if you have proprioception coming in and if you can actually stay balanced when you close your eyes. But we can look at this a little bit more clinically or a little bit more subtle way to find out these lesions or functional disconnections that she might have. Okay, so what I'm going to have her do, put her feet together and just relax and close her eyes. I want to make sure that she's not falling. And she's pretty well balanced right now. Okay, go ahead and open your eyes. And what you want to be looking at here is the very initial sway. Did they sway to the right, to the left, to the back, to the front, or in a diagonal way? And you wanna write that down. Oftentimes, if the patient has the initial sway, it says the tiny one to the back and to the left, they might compensate a lot by going straight to the right, diagonally. So you want to make sure you notice that initial sway, because that might be the one that's their pathology. And same thing here. This is also a big vestibular test. But we're also looking at the proprioception, and in this case as well, we usually say that the patient might fall to the side of the hypofunctioning cerebellum. Okay. Next, we're going to look at the rapid alternating movements. So if you're going to be testing more of the midline cerebellum, we're going to do the rapid alternating test here. So go ahead and do pronation and supination as fast as you can. And then you observe from side to side if there are any off-axis inaccurate movements. Okay? She looked pretty good to me. I didn't get to see really well, but that's what you're going to be looking at, okay? And then also you want to look at facial tone. You want to look at if there's any limbic escapes. Are they trying to compensate from one side or the other, etc. because you're looking at the complete system. And that's why we always talk about try to look with your peripheral vision. Don't try to look at the central vision into their eyes, but try to look at their whole body. And it's going to give you a lot more to look at, okay? Next, we're going to do rapid alternating movement in here. So I'm going to have her place her hands like this, and then same thing, supination, pronation as fast as you can. Perfect. Relax. So this one is more for the interposed nuclei, like we talked about, the embolus nucleus, okay, for the elbows. Then what we're going to have you do is go ahead and place your arms like this, and then play the piano as fast as you can. And you want to involve all the digits, even the thumbs, as fast as you can. We can see she has some limbic escapes here. She's laughing here. Okay, she's having some trouble here on the left as well. You just can't do it. Too funny. But that's where you want to be looking at, okay? So oftentimes what you see is the patient has the fingers will move continuously together. So like it's really hard to mimic this, but they might move these two fingers together or these three together and they don't even notice it, okay? And same thing, you compare bilaterally and you look at everything. Next, we're going to do finger to nose. So what I'm going to have her do is go ahead and place your arms out like this. And I'm going to touch one of your fingers, and then I want you to go ahead and touch your nose like this with that finger that I touch you on. So go ahead and close your eyes. So first of all, you want to look that they're actually touching their nose with the appropriate finger, the accurate finger that you actually touched on. Perfect. Thank you. And you also want to look at the smoothness of the movement. Is there what we already talked about? Is she undershooting? Is she overshooting? Is she hitting herself in the face? Is she actually touching the target, her nose? Does she know where her nose is in space, and does she have a proper parietal map of her face? That could be a thing too. But specifically looking at the movement, how accurate the movement is there any intention tremor at the end? Do they do this? That's a classic cerebellar sign. When you get closer to the object or the target, you start tremoring at the end. And that's called the intention tremor. Okay, next we're going to do heel to shin. So go ahead and lay down, face up. What I want you to do is I just want you to place your heel on your knee and slide down all the way to your big thumb along your shin. Perfect. Now do the other side. Perfect. And you want to go all the way from the knee to the shin and to the big toe. Okay. And you're trying to look if the patient is going to be accurate or if they're going to stay on the shin or if they have trouble from going, staying on the shin and they go like this. Okay. This is often what you see with the cerebellar lesion. And same thing here, if you're looking at a right cerebellar lesion and she would perform the right one, you might see those things appear. And now we're looking at the left cerebellum. Okay? Perfect. Then go ahead and stand up. And next one on the list, we got the oculomotor function. We're going to talk about that more in the vestibular, the VOR week. And we're going to have 2 weeks for that. And we're really going to pound on those eye movements. Okay. So go ahead and try these with your partner and try to find whether you find a right cerebellar or a left cerebellar findings. You might have both. Okay. The system is really complex. You might not always get ipsilateral findings and always following this pathway we always talk about, this cerebello-contralateral frontal lobe back to the same side of the cerebellum. You might find different findings, and then you have to look at your entire neuro exam and figure out what's happening in that patient's nervous system so we can give the most appropriate therapy for them. Okay, thanks.
So after you've gone through all of these tests with your partner, do something to recheck. So write down what, if they were weak, you say all on the right side, and they had a lot of right cerebellar findings, try something to activate that right cerebellum, like we talked about last week with the complex movement. So do a complex movement with their arm or maybe their arm and leg, and try that, and then retest the tests that they had trouble with and see if it makes a difference. Because usually with the therapies, if you use, you can also use them to stimulate the cerebellum, so the test can also be the therapies, but try something to just do that test retest. That's what we really want to get used to and start doing every day when we're practicing with our patients.
Perfect. And something else you can do is adjust them, okay? So adjust them on the side of weak cerebellum. And when we talked about week 1 about spatial summation and temporal summation, you could try temporal summation, for example. So adjust your fingers on this side of the body, then elbow, and then shoulder, and then recheck these findings, these tests, and if they got better or if they got worse. If they got worse, then you know you might be dealing with something else and you might want to try the other side, or you might have to think about other structures in the brain that's going on in there, okay? So go ahead and play with this. It's all about pre and post checking, you know? Perfect. So that's it.